Welcome to the lecture series on control of gene expression. I'm Janine Post of the Developmental Bioengineering Group at the University of Twente. In this third lecture, I will talk about how transcription regulation results in the generation of specialized cell types. Illustrations in this video are based on the book Essential Cell Biology, Chapter 8. In this series, I give 4 short lectures on gene expression control. In this lecture, I will focus on the cell type-specific regulation of gene expression. Tissues consist of many different cell types that work together to maintain specific functions. In the schematic shown here, a part of the gut is visible, with different cell types that function together in the processing of food. So the real question is, how is the development of these different cell types regulated? Well, the specialization of cells is regulated by the activity of cell type-specific transcription regulators. When we look at the overall picture of the regulation of gene expression, it looks something like this. Signals from the extracellular region bind to receptors on the cell surface. This leads to the simultaneous activation of many signaling pathways and intracellular signaling molecules. Ultimately, this leads to activation of transcription regulators that translocate to the nucleus. In the nucleus, they bind to specific regulatory sequences in the DNA, where each transcriptional regulator binds to its own corresponding regulatory sequence, which we also call response element. And together, the transcription regulators regulate gene expression. The response element shown here is bound by the transcription factor NANOG. So, if you remember, the promoter region, including the theta box, is recognized by general transcription factors that bind and phosphorylate RNA polymerase II to start transcription. But here we want to discuss regulation of gene expression by tissue-specific transcription regulators. These tissue-specific regulators can bind at a large distance from the transcription start site by binding to a distant enhancer, here shown in green. This binding attracts a complex of proteins, including RNA polymerase and the general transcription factors that bind to the promoter. The DNA loops so that there's contact between the activator and the transcription initiation complex that is bound to the promoter. Here you can see that a large protein complex called mediator in purple serves as a go-between. The broken stretch of DNA signifies that segments of DNA between the enhancer and the start of transcriptions vary in length, sometimes reaching tens of thousands of nucleotide pairs. So what is this mediator? What is illustrated here as the mediator is in fact a complex of many different types of proteins, including chromatin-modifying enzymes such as histone acetyltransferases, chromatin remodeling complex proteins, and docking proteins for general and specific transcription factors. So how does that work? As you know, DNA is wound around an octamer of histone proteins that form a bead on a string, which you can see by electron microscopy. This is further packaged in a 30 nanometer fiber, also visible by electron microscopy, and that folds into loops. This higher organization of the DNA ensures a precisely controlled nuclear architecture that prevents tangling of DNA, but it also controls cell function, both by modification of the DNA, for example DNA methylation, as well as at the level of histone, for example histone tail acetylation. So what is then necessary for transcription to occur? The recruitment of histone-modifying enzymes, so as histone acetyltransferases, or HATs, add acetyl groups to specific histones, which can then serve as binding sites for proteins that stimulate transcription initiation. In addition, chromatin remodeling complexes render the DNA packaged in nucleosomes more accessible to other proteins in the cell, including those required for transcription initiation. Here is shown the increased exposure of the theta box. Therefore, the proteins regulating this modification at the level of chromatin are found in the mediator complex. Specialized proteins, shown in green, hold chromosomal DNA in loops to make sure each gene can associate with its proper enhancer, shown in blue. The loops, sometimes called topological associated domains, or TADs, range in size between thousands and millions of nucleotide pairs and are typically much larger than the loops that form between regulatory sequences and promoters. So how are the different cell types within tissues formed? Cells must be able to turn genes on and off in response to signals in their environment. In multicellular organisms, there's additional transcriptional control. This allows formation of organized arrays of differentiated cell types, shown here. This has everything to do with lineage commitment. Once a cell becomes committed to a specific cell type, the choice in cell fate needs to be maintained through subsequent cell divisions, indicating that changes in gene transcription must be remembered by the cell. This so-called cell memory is maintained by combinatorial control, positive feedback loops from transcription factors, and epigenetic inheritance, for example, DNA methylation. Let's look at those processes in more detail. Eukaryotic genes are controlled by combinations of transcriptional regulators that work together as a committee to control gene expression of a certain eukaryotic gene. In contrast to the general transcription factors that assemble at the promoter, and are the same for all genes transcribed by RNA polymerase II, the transcription regulators, or transcription factors, and the location of their DNA binding sites relative to the promoters are different for different genes. These regulators, along with chromatin-modifying proteins, are assembled at the promoter by the mediator. The effects of multiple transcriptional regulators combine to determine the final rate of transcription initiation. The spacer DNA that separate the regulatory DNA sequences are not recognized by any transcription regulator. So ultimately, many different transcription regulators are necessary to initiate transcription of certain genes. A single transcription regulator can coordinate the expression of many different genes, which is illustrated in this example for the cortisol receptor on 3 different genes, each of which has a different activator protein bound to its respective regulatory DNA sequences. These bound proteins are not sufficient on their own to activate transcription efficiently. As shown on the right, the presence of cortisol is necessary to activate an additional transcription regulator, the cortisol receptor complex, which binds to the same regulatory DNA sequence in each gene. The activated cortisol receptor completes the combination of transcription regulator required for an efficient initiation of transcription, and all 3 genes are now switched on as a set. Combinations of a few transcription regulators can generate many cell types during development. In this simple scheme, a decision to make a new transcription regulator, shown as a numbered circle, is made after each cell division. Repetition of this simple rule can generate 8 cell types, A through H, using only 3 transcription regulators, 1 in green, 2 in red, and 3 in yellow. Each of these hypothetical cell types would then express different sets of genes, as dictated by the combination of transcription regulator that each cell type produces. Here's another example of combinatorial control. A set of 3 transcription regulators forms the regulatory network that specifies an embryonic stem cell. These 3 transcription regulators, KLF4, OCT4, and SOX2, are shown in large colored circles. The green dots are the genes whose regulatory sequences contain binding sites for each of these regulators. The lines that link each regulator to a gene represent the binding of the regulator to the regulatory region of the gene. Here you can see groups of genes that are regulated by only 1 of these transcription factors, by 2 of the transcription factors, or by all 3. On the right here is indicated that these three transcription regulators also control their own expression. As shown here, each regulator binds to the regulatory region of its own gene, as indicated by the feedback loops in red. In addition, the regulators also bind to each other's regulatory regions, as shown in blue. These red loops are an example of positive feedback loops, a common form of regulation. Master transcription factors can regulate the direct formation of an entire organ. For example, when the Drosophila eye gene is artificially induced in the precursor cells of the leg, an eye will form on the fly's leg. The experimentally induced organ appears to be structurally normal, containing the various types of cells found in a typical fly eye. It does not, however, communicate with the fly's brain. A small number of transcription regulators can convert one differentiated cell type directly into another. In the experiment shown here, liver cells are grown in culture and were converted into neuronal cells by artificial introduction of 3 nerve-specific transcription factors. Such interconversion would never take place during normal development. The result shown here depends on the authors of the paper, Marrow et al., expressing several nerve-specific regulators in liver cells, where these regulators would, during normal development, be tightly shut off by epigenetic regulation. A combination of transcriptional regulators can induce a differentiated cell to dedifferentiate into a pluripotent, induced pluripotent stem cell, or iPS cell. The artificial expression of a set of 3 genes, KLF4, OCT4, and SOX2, which contain the pluripotency network, can reprogram a fibroblast into a pluripotent stem cell with embryonic stem cell-like properties. Like embryonic stem cells, such iPS cells can proliferate indefinitely in culture and can be stimulated by the appropriate extracellular signal molecules to differentiate into almost any cell type of the body. A positive feedback loop can generate cell memory. In this example, protein A is a master transcription regulator that activates the transcription of its own gene, as well as other cell type specific genes, which are not shown in this image. All of the descendants of the original cell will therefore remember that a progenitor cell had experienced a transient signal that initiated production of protein A. Another type of cell memory is provided by DNA methylation patterns that can be faithfully inherited when the cell divides. This is a type of epigenetic memory. The memory is not encoded by the order of the DNA bases ATCG, but by methylation of the C base by an enzyme called a maintenance methyltransferase. This methyltransferase guarantees that once a pattern of DNA methylation has been established, it is inherited by newly made DNA. Immediately after DNA replication, each daughter double helix will contain one methylated DNA strand, inherited from the parent double helix, and one unmethylated, newly synthesized strand. The maintenance methyltransferase interacts with these hybrid double helices and methylates only those CG sequences that are base paired with a CG sequence that is already methylated, thereby maintaining memory. Histone modifications can also be inherited by daughter chromosomes. Shown in this model, when a chromosome is replicated, its resident histones are distributed more or less randomly to each of the daughter DNA double helices. Thus, each daughter's chromosome will inherit about half of its parent's collection of modified histones. The remaining stretches of DNA receive newly synthesized, not yet modified histones. If the enzymes responsible for each type of modification bind to the specific modification they create, they can catalyze the filling in of this modification on the new histone. This cycle of modification and recognition can restore the parental histone modification pattern and ultimately allow the inheritance of the parental chromatin structure. The maintenance of the condensed heterochromatin is regulated by these types of epigenetic regulation, and together these processes maintain cell fate commitment and generation of specific cell types. Thank you for watching. If you have any questions, please email me. In the next lecture, I will discuss different types of post-transcriptional controls.